Suman sir, is my voice audible? Hello?
Audible, sir.
Okay.
Good morning, sir.
Is my voice audible?
Hello, good morning, sir. Good morning, sir.
Yeah, your voice is audible.
Good morning to all. Say a warm welcome to all the participants who have registered for this 5-day FDP on innovative methods in analytical sciences, organized by Sri Vasavi Institute of Pharmaceutical Sciences, Tadepalligudem. I welcome Dr. V. Bhaskar Raju garu, Principal, Sri Vasavi Institute of Pharmaceutical Sciences, Dr. A. Krishnamanjari Pawar Madam, Associate Professor, Department of Pharmaceutical Analysis, Andhra University College of Pharmaceutical Sciences, Vishakhapatnam, and she is the today's speaker. Warm welcome to all the participants for this FDP. So now I request Dr. V. Bhaskar Raju garu to address the participants.
So thank you, Suman sir. Yeah, good morning to all the participants and today's resource person, Dr. A. Krishnamanjari Pawar Madam. So she is the Associate Professor and HOD Department of Pharmaceutical Analysis, Andhra University, Vishakhapatnam. And good morning, Madam, and good morning to all the participants and welcome to the day 4 of our EFDP and on innovative methods in analytical sciences. On behalf of our faculty and students, I welcome our participants and resource person for today's FDP. And we are very excited and delighted to hear from our resource person a valuable and informative session, which should be very useful for our participants. So thank you for accepting our invitation, Madam, and sparing your valuable time for the benefit of our participants. And now I request our technical director, Sir Sri C.H. Apparao garu, to address the participants and our resource persons.
Good morning, everybody. I'm really happy that we are all participating as part of this 5-day session. Today is the 5th day. We welcome the speaker for today to really, I think what I heard from everybody is like he's a very information person, Mr. Power. Sir, we really welcome you to our college and be part of this program. Please, I think a lot of people are waiting to hear from you. I request you to be part of it. And then as part of our college, we welcome you to WASPI Pharmaceutical College. So with this, I really thank for giving your valuable time and being part of this. Welcome, sir.
Thank you, sir. And now I request our Assistant Professor, Department of Pharmaceutical Analysis, Mr. Sharma, to give a brief introduction about our research person, Dr. Pawar Madam, followed by the presentation. Sharma, please continue.
Thank you, sir. Good morning, everyone. Good morning, ma'am. It's my privilege to introduce today's resource person, Dr. A. Krishnamanjari Power ma'am, Associate Professor, Member of Executive Council, Andhra University of Pharmaceutical Sciences, Andhra University, Visakhapatnam. Dr. A. Krishnamanjari Power ma'am completed B.Pharmacy from Kakatiya University in the year 2002 and M.Pharmacy from Andhra University in the year 2005. She completed PhD from Andhra University in the year 2012. Dr. A. Krishnamanjari Power ma'am had teaching experience of more than 19 years and she received various awards like Pharmacy, like Pharma Teacher of the Year 2022 by Pharmacy Teachers Trust and Dr. Sarvepalli Radhakrishnan Award for Best Academician of the Year 2022 by Andhra University, Visakhapatnam. IMRF Best Scientist Award from International Multidisciplinary Research Foundation in 2019 and Outstanding Researcher in Pharmaceutical Analysis Award from Venus International Foundation in the year 2018. Madam also received 4 more professional awards from various international and national organizations. Dr. A. Krishnamanjari Power ma'am has guided 69 M.Pharmacy students and 15 PhD research scholars. And at present, ma'am is guiding 23 PhD research scholars. She had 71 national and international publications, and her interest in area of research is analysis of food and herbal products and impurity profiling of drugs and pharmaceuticals. So I kindly request Dr. A. Krishnamanjari Power ma'am to start this session. Thank you.
Thank you so much. Sharma garu,
Yes, ma'am.
am I audible and visible?
You're audible, ma'am, but your video is not clear, ma'am.
Please check now.
Sorry, ma'am, video not visible, ma'am. Audio is clear, ma'am.
Okay. Can you give me a few minutes, please?
Yes, ma'am.
Hello, sir. Sharma garu, now am I audible and visible?
Yes, ma'am.
Yes, madam. Yes, madam.
Yes, everything is set.
Yes, madam.
Yes,
Thank
ma'am.
you so much. Sorry for the interruption. There was some technical issues. So let me share my screen. Could you able to see my slides, please?
Yes, madam.
Thank you so much. So namaste everyone. Welcome to 5th day of faculty development program on innovative methods in analytical techniques conducted by Sri Vasavi Institute of Pharmaceutical Sciences, Tadepalligudem. I appreciate wholeheartedly the management of SVIPS, Sri Subbarao Garu, Secretary Correspondent, for facilitating these kinds of professional activities in your esteemed organization. I would also like to praise the efforts of Principal Dr. Bhaskar Raj Garu and Organizing Secretary K. Sumant Garu for taking this initiative and working in hand to hand with other faculty members and non-teaching staff of your college. And I also like to thank all of you for inviting me for this lecture, and I'm so proud to be a part of this program. And it's always proud to see the youngsters on board in organizing committee. And I appreciate Kalyani, Pallavi, Yamini, and Sharma, the youngsters who are actively involved in organizing this FDP. Good job, guys. Let's begin our journey with a great quotation said by Mark Twain, an American writer. The secret to getting ahead is getting started. So, my dear participants, let's cheer each other for getting started in this new journey. You are going to see the new world, new challenges in your life. So get ready to face all those things. In this current digital world, Make in India, NEP 2020, entrepreneurial era, we are all so enthusiastic. At the same time, we are in some stress to understand all these technologies. So, to understand all these things, we are starting our journey. So, let's all cheer up once again. Today, I am going to talk about recent developments in analytical science. I am Dr. A. Krishnamanjari Pawar, Associate Professor, AU College of Pharmaceutical Sciences, Andhra University, Visakhapatnam. So, when we look into the contents of today's topic, right from the top left side, what are analytical sciences? Need for innovative methods in analytical science. importance of innovation in analytical science, innovative methods. In that, we are going to discuss about imaging techniques, biosensors and bioanalytical methods, chromatography, mass spectrometry, spectroscopy, and applications of AI in analytical science, challenges and future aspects, way to overcome challenges in analytical field. These are all the contents of our topics. What is analytical science? So analytical science is a branch of science that focuses on the development and application of methods to analyze and measure substances in order to understand their properties and interactions, quantity, safety, and effectiveness of the samples. Here, a substance may be a drug, pharmaceutical, chemical entity, cosmetic, Ayurvedic product, or any other product. Whatever it may be the product, but we have to analyze the sample and we have to report it according to the needs of the human being. What is the importance of innovation in analytical science? If we broadly classify the innovation in analytical science, the importance goes to first advancement of research and enhanced precision and accuracy. So here, the innovation in analytical science plays a crucial role in advancing the research, research capabilities. It enables a scientist to develop new techniques and methodologies that led to breakthroughs in various fields such as medicine, environmental studies, and material sciences. By embracing the innovative methods, analytical scientists can achieve higher the precision, accuracy in their measurements and analysis. This is essential for ensuring the reliability of experimental results and making informed decisions based on data. So, the importance of innovative analytical sciences, what is the cost and time efficiency? If we have a glance on that, innovation in analytical science often leads to the development of efficient and cost-effective techniques. This can significantly reduce the time and resources required for conducting complex analysis, making the research more accessible and impactful. Then addressing the complex challenges. So the new and innovative methods equipped with the tools are useful to tackle the complex analytical challenges, whether it's dealing with intricate sample matrices or identifying the trace contaminants. So innovation is essential for overcoming the obstacles in the analytical science. What is the need for innovative methods in analytical science? So as we all are aware that we have been listening and watching the growing needs of a human being in all the aspects of life. Development of personalized medicine, newer technologies, portable devices, non-invasive technologies. So what we need is new innovative methods. Because of all these things, there is a growing complexity of samples as well as the analysis requirements. We have to monitor quality control. Analytical methods are often used for monitoring the quality control purposes. So emerging analytical needs, because emerging need of a human being in each and every sector of the industry, and advances in technology. As technology continues to evolve, there is a constant need to develop new methods that can harness these advancements. So we need innovative methods. What are the benefits and applications of innovative analytical methods? So, by developing and adopting new innovative analytical methods, we can improve the sensitivity, selectivity, speed, accuracy, reliability, and we can adapt to these innovative analytical methods to the newer applications. Then let's talk about cutting edge analytical techniques. So you can see the new cutting edge analytical techniques like virtual AR, a gas chromatography which can be fit into a palm, flow cytometry, lab on chip, ion mobility mass spectrometry. So all these things are new cutting edge analytical techniques which have arrived in the world. So to know much about the cutting edge analytical techniques in the new era, we can broadly classify all these things and we can fit enough all the newer technologies into these categories. First is imaging techniques. The second one is biosensors and bioanalytical methods. The third one is chromatography. The fourth one is mass spectrometry. And the fifth one is spectroscopy. So here, if we have a glance on the advances in innovation in analysis, so miniaturization and portability has increased. Integration of multiple techniques has tremendously increased. Artificial intelligence and machine learning in data analysis is increased. Automation and high throughput analysis is also increased. So let me tell you the importance of newer technologies, how they have emerged into our lives in recent times after after COVID era, is the top 10 imaging technologies in 2024. These are the top 10 technologies which are utilized by the mentioned companies in 2024. So these technologies include computer vision, connected imaging systems, generative AI, hyperspectral imaging, high-speed imaging, XR visualization, LiDAR, 3D imaging, nuclear imaging, 4D imaging. So see how the world is changing so fast. So what are the imaging techniques? So the imaging techniques refers to various methods and technologies used to create visual representation of objects or structures that are otherwise not visible to the naked eye. These techniques are commonly used in fields such as medicine, biology, astronomy, material science, among others. Here are some commonly used imaging techniques like positron emission tomography, magnetic resonance imaging, single photon emission tomography, optical imaging, and others can be detected by the detectors, X-rays, CT computed tomography, MRI, ultrasound. So the types of imaging techniques can be like this: optical microscopy, 3D imaging innovations, extended reality visualization, hot stage microscopy, laser scattering. So if we see what is optical microscopy, so this technique employs to closely view a sample through the magnification of lens with visible light. So recently the performance of optical microscopy has been enhanced the emergence of advanced devices like ferroelectric liquid crystal spatial light modulators, SLM, 2K or 4K digital micro mirror devices, high power LED light, resonance scanning, galvanometers, sensitive scientific CMOs, cameras. So all these new devices has made optical microscopy to increase its sensitivity from 200 nanometers to 500 nanometer resolution. Magnification power has increased up to 1000x. And in terms of performance, we can observe high imaging quality and high resolution power. When we look into the applications of optical microscopy, we can document the optical properties like color, size, identification of the particle, and the differentiation between amorphous and crystalline material, particle distribution within the matrix, and exact determination of melting point and simultaneous observation of melting, evaporation, and crystallization behavior. So, coming to the second technology, 3D imaging innovation. So here, the 3D imaging innovation advances in like visualization of a component, visualization of a sample, visualization of a particle. So advances in 3D imaging includes better image quality, functional data utilization, and enhanced quantitative analysis. So integration of AI and machine learning into imaging equipment and analytical tools transforms 4D imaging for dynamic processes, not for static processes. So here, the 4D imaging captures the changes over time and adding the temporal dimensions to spatial data.
அப்புறம் இதெல்லாம் பண்ணிட்டு செமஸ்டர் போட்ட ஆமா ஃபீஸ் கட்டிடுவாங்க
Sharma garu, there is a disturbance from some other
படிங்க எங்கப்பா நான் தான் போகணுமா கொஞ்சம் போயிருக்கோம்டாமா நீ பாரு அவ இளைச்சி
Participants, please unmute yourself.
டெய்லி நடந்து நடந்து வந்து நான் இளைச்சிட்டேன் மேம் மேம் திரும்பி ஃபோன் பண்ணி
please control your speaker with your central command.
மேம் இவளை முடிச்சுடுங்க
யார அவன் அவங்க பஞ்சாயத்தெல்லாம் முடியாதுமா இதெல்லாம் அவங்க பஞ்சாயத்து கிளப்பணும் ரெடியா இருக்கிறாங்க அவங்கள எல்லாம் முடிச்சு
அப்புறம் என்கிட்ட சொல்லுவாங்க மேம் நான் நம்பர் நான் நம்பர் கேட்டேன் நீங்க தான் தரல அப்படிமா
எதுக்கா நம்பர் அனுப்பிச்சு அவங்க அம்மா நம்பர் இருக்கா நான் நம்பர் அனுப்பிச்சிடுவேன்
சரி மேம் நாங்க அந்த சிஸ்டத்துக்கே போறோம் கேட்டு போயில என்ன ப்ராசஸ் அதுதான் நேத்து போட்டாங்க வாங்க மேடம் இங்க பேசிட்டு இந்த ஃபைல நீங்க பேசுறது நம்பர் சொல்றீங்களா மேம் நோட் பண்ணி அத வந்து எஜுகேட்டர் நம்பரை நீங்க கால் பண்ணி பேசிக்கோங்கன்னு சொல்றதா மேம்
இவங்களுக்கு ஃபோன் பண்ணி பேச சொல்லுங்க அவங்க தான் அவங்க கட்டிச்சு போட்டவங்க என்ன அவங்க பேசி அவங்க அப்புறம் என்கிட்ட
So here, the 3D imaging technique with the including AI and machine learning, the 3D has become into 4D, and the 4D imaging captures the changes over time and adding the temporal dimensions to spatial data. Sharma garu, am I audible and visible?
Yes, madam.
Okay, thank you. So it's a crucial study for dynamic processes in the fields like biology, material science, and geology. Not in the static process. In the dynamic process, we can take the 4D imaging. That gives a tremendous information about the picture. Then the nuclear imaging techniques. So the nuclear imaging techniques such as PET and SPECT provides the functional information about the tissues and organs. Both the qualitative and quantitative particle analysis can be done by all these techniques. So the nuclear imaging such as PET and SPECT innovations enhances the resolution, sensitivity, and specificity for better disease diagnosis. Then coming to the point, extended reality visualization. So the XR technologies include augmented reality and virtual reality, which delivers the immersive experiences. In imaging, XR enhances the visualization, training, and data analysis. See, you can see in the right side picture, the 4D MRI provides a non-invasive way to visualize even the blood flow in arteries. So this extended reality can be like augmented reality, mixed reality, and virtual reality. These are all come under one umbrella, XR Tech. So the umbrella term that encompasses any sort of technology that alters the reality by adding the digital elements to the physical or real world environment by any extent. So in the augmented reality, a digital layer over the physical element. Say, suppose this is a physical element, over this, there will be a AR reality, and we can see what it is. And in mixed reality, the digital elements can interact with the physical elements and gives us crystal clear clarity about this physical element. And in virtual reality, we need not to have any physical element, and it is totally immersive digital environment. So we are all very aware of this virtual reality in our real life, as our kids are daily playing with VRs, and shopping malls are, making money out of this technology. Then the video, you can see how these technologies are helpful to the profession. The video depicting this augmented reality visualization, you can see once. A surgical AR vision as an augmented reality app for medical professional. It transforms the 2D medical imaging data into interactive 3D visualization, giving the surgeons the vital anatomical insights in real time. This innovative technology allows the surgeons to view the crucial anatomical details without shifting the focus away from the patient, and they can do their operations very precisely. Coming back to the slides. The fourth topic is the hot stage microscopy. So here, hot stage microscopy is defined as the thermal analysis technique that combines the properties of thermal analysis and microscopy. So this is a powerful hyphenated technique which is widely used to visualize and examine all kinds of thermal transitions like melting point, crystallization, solid state transitions, and decompositions in real time. So combination of microscopy and thermal analysis is to enable the study of material as a function of temperature and time. So it allows the researchers to observe the behavior in real time and enables the melting temperatures and analysis of the samples in crystalline structure as it undergoes the transition from solid to liquid state. You can observe the hot stage microscopy when determining the melting temperatures and behaviors from left side to right side, the changes and real-time analysis. Then coming to hot stage microscopy and digital thermal analysis, HSM and DTA. Coupling the thermal analysis with microscopy for the solid state characterization for materials as a function of temperature and time is HSM DTA. So in pharmaceuticals, HSM is used to support DSC and TGA observations and to detect small changes in the sample that may be missed by DSC and TGA during the thermal experiment. Then coming to the point, laser scattering. So the laser scattering technique uses a laser beam as a probe and measures the scattering of the light by particles or surfaces in a medium. So the laser beam is directed towards the sample and the scattered light by electrons is collected and analyzed. So some of the electrons are rejected. So based upon the rejected electrons, the shape of the part or shape of the organ will be identified easily. And the technique can provide information about the size, shape, concentration, and refractive index of the scattering particle. The laser scattering is commonly used in various fields such as physics, chemistry, biology, environmental science, material science for characterization and analysis of samples. So if you can classify it, it can be like dynamic light scattering and static light scattering. Dynamic measures the fluctuation changes on the intensity of the scattered light, and static measures the total intensity of scattered light. Coming to the advantages of laser scattering, high resolution scalability is increased with this technology, and speed enables the scanning in a production level, and the flexibility allows quick changes to patterning the parameters, and reduced process steps because it potentially streamlines the manufacturing workflows. So we can include the laser scattering in the workflows. We need not to take the sample from the place of the production and go to somewhere else. So we can inline join this technology. High yield precision contributes to defect-free production. Compatibility works with various substrate materials. Then laser scattering applications. So with this laser scattering, we can identify the particles size, particle shape in sprays, powders, emulsions, suspensions, etc. Then the applications of laser scattering in research and development, the particle size analysis, protein aggregation, dynamic light scattering, environmental monitoring can be done, nanoparticle characterization can be done, colloidal stability of the analysis can be seen in this suspensions and emulsions, polymer characterization can be done in material science, biomedical diagnostics, detect the biomolecules and cells for the disease diagnosis, rheological properties can be identified, and drug delivery systems can analyze drug delivery formulations for efficiency and stability. Then coming to the point of biosensors and bioanalytical methods. So far we have seen about the imaging techniques, and we are coming to the second topic, biosensors and bioanalytical methods. So, as the technology is advancing in the recent years, especially during the COVID and post-COVID, these usage of biosensors is tremendously increasing in the medical field. So, biosensors and bioanalytical methods, what are they? Let us have a glance. Biosensors are analytical devices that convert a biological response into an electrical signal. Quintessentially, biosensors are must be highly specific, independent of physical parameters such as pH and temperature, and should be reusable. This is the character of a biosensor. Coming to bioanalytical methods, it is a quantitative measurement of a concentration of a drug and or its metabolite or a biomarker in biological fluid such as blood, plasma, urine, serum, etc., or in tissue extracts. So the advantages of biosensors: high sensitivity, detects low concentration of analytes even in complex samples because all the biosensors are very teeny in size. Then rapid response, provides quick results, facilitating the real-time monitoring and diagnostics. Specific detection offers selective detection of target analytes, minimizes the false positives, and miniaturization of the technology, portability, and real-time analysis, point-of-care applications for on-site testing. This is the advantage of biosensor. Low cost and versatility, adaptable to various analytes, sample matrices, enhancing the applicability as it is going to analyze the single analyte. Then automation, compatible with automated systems, reducing the manual labor and errors. So long-term stability maintains the performance over extended periods, ensuring the reliability. Ease of use, so use-friendly interfaces and simple operations suitable for non-experts. Simply we can use by downloading an app in our mobile and doing whatever the parameters we need. We can enter into that app and we can get the results. Potential for integration can be integrated with electronics and microfluidics for enhanced functionality. Then the various biosensors are, they detect specific biological molecules such as glucose, protein, or DNA, and their interactions into measurable signals. So the recent developments include glucose biosensors, fluorescent biosensors, antibody-based sensors, enzyme-based sensors. So what are microfluidics? So microfluidics is the science and technology of manipulating and controlling. Just a minute. Tiny amounts of fluid, typically in the microliter or nanoliter range, within a small channel or a small device, micro level. So microfluidics refers to the study and manipulation of small volumes of fluids, typically on the scale of microliters to picoliters within the microscale channels and structures. So it involves the design, fabrication, and application of the devices, often referred to as microfluidic chips or lab-on-chip system that can precisely control the flow, mixing, and reactions of fluids at a microscale. So the microfluidic technology enables a wide range of applications in the field such as analytical chemistry, biotechnology, biomedical engineering, material science, and it offers the advantages such as reduced sample, very little amount of the sample we need here. Reagent consumption is also less, and faster reaction times, enhanced sensitivity, and ability to integrate with multiple laboratory functions onto a single chip. Wearable microfluidic devices for biofluid analysis. Wearable devices now integrate microfluidics and smart technologies such as artificial intelligence and machine learning for real-time analysis of biofluids like sweat, interstitial fluid, tears, saliva, own fluid, and urine. So these advancements enables the accurate and non-invasive health monitoring and diagnosis. So wearable devices are gaining popularity in the field of health monitoring and diagnosis and drug delivery. So the recent advances in wearable technology have enabled real-time analysis of biofluids such as sweat, interstitial fluid, tears, saliva, own fluid, and urine. So the integration of microfluidics and emerging technologies such as AI, ML, IoT into wearable devices offers the great potential to accurate and non-invasive monitoring and diagnosis. So you can see these are the wearable sweat monitoring patch, disposable strips we can use like this in the left side in A picture. So you can use these chips straight away, take your sample and drop it in the picogram level and you can get the results. Then three-dimensional microfluidic devices. These devices refers to a microfluidic system that generally incorporates three-dimensional structures such as channels, chambers, and other geometrical elements that extend in three spatial dimensions rather than being confined to a single plane or layer. So it not only determines the sample type, but also enables us to determine the structure and shape of the sample in three dimensions. So that this 3D cell culture micro devices aiming to reduce the key functions of living organs on a computer chip. So the microfluidic devices are more efficient than conventional cell culture techniques since they are able to mimic micro environment as well as their influence on organ function. So we can take the cell of our organ, and if we can use this multi-dimensional microfluidic device data, that it will tell us, do you have the cancer or not, whether it has a spread or not, what is the progress of it, how your organ is working, what may be the probable problems in future for that organ. So this kind of informations we can know by three-dimensional microfluidic devices information. So these intricate corkscrew-like designs allows the precise fluid control, efficient particle separation, and reagent mixing. They find the applications in medicine, biology, and chemistry. So microfluidic systems manipulate small volumes of fluids to perform the automated task with applications ranging from chemical synthesis to biomedical diagnosis. The advent of low-cost 3D printers has revolutionized the development of microfluidic systems. So the advantage of three-dimensional system microfluidic devices are enhanced control and manipulation of the fluids. So you can manipulate, you can shut up your fluid influences on the sample. Then increase the efficiency and performance, reduce the sample and reagent volumes, integrate the multiple functions in a compact design, and improve the sensitivity and accuracy as well, and enable the miniaturization, portability, and point of care applications. Then coming to the point of advances in sensor technology for biomedical applications. So the novel developments in biosensor include fiber optic sensors, microelectrochemical sensors, microelectromechanical systems, MEMS, flexible sensors, and wearable sensors. So these technologies enhance the real-time monitoring, diagnosis, and personalized healthcare. So fiber optic sensors. These fiber optic sensors offers the advantages like high sensitivity, immunity to electromagnetic interferences, compatibility with minimally invasive procedures. They find applications in endoscopy, intravascular sensing, and optical coherence tomography. The flexible sensors confined to irregular surfaces and can be integrated to wearable devices, skin health monitoring, motion tracking, wearable ECG monitors. So then the MEMS devices have revolutionized precision surgery, diagnostics, and therapeutic systems. For example, bio-inspired robotic fingers equipped with tissue softness characterization sensors for precise pressure measurement and during the surgery for the movements of the fingers. Blood cell counting sensors, so rapidly assess the blood cell levels. Acoustic sensors, localized 2D sounds, vibrations can be known by these sensors. Insects like flying robot sensors. Suppose we have mines, we cannot take all these technologies there. So we can take these flying kinds of robot sensor and get the information. Then the bio-inspired robotic fingers, what they do is grasping and manipulating the object, detecting the touch and pressure, providing the haptic feedback, enhancing the dexterity and precision. Blood cell counting sensor does the diagnosis of medical conditions, monitoring the disease progression, evaluation of the treatment effectiveness, how the blood cells are improved, and identification of abnormal cell population, the cancer identifications and all. The acoustic sensors are used to detect and measure sound waves and vibrations. They are used in microphones, hydrophones, industrial machinery monitoring, automotive noise cancellation, medical imaging, and more. Insect-like flying robot sensors track the insect movement and migration patterns, monitor the population dynamics and abundance, detect and identify the insect pests in crops and urban areas, studying the insect behavior and responses to environmental conditions, developing the strategies for insect control and management can be done by using these kinds of sensors. So, what are the challenges in these techniques? Are the sample contamination. There is a high potential risk of contamination during the invasive procedures, as they simply usually handled by the individuals. And accuracy concerns, limitations in the accuracy of sensor measurements, and then complex data interpretation. Challenges in interpreting the data collected from invasive techniques and sensors is typical for the individuals. We need expertism. Then the third topic, chromatography. You all are very aware of chromatography, so I don't want to talk about the basics. So types of chromatography, I have depicted here around 10 techniques, but we have plenty of other techniques. So the importance of chromatography in an analytical science is because of its precise separation. Chromatography enables the precise separation and identification of complex mixtures, making it invaluable in analytical science and diversified applications. From pharmaceutical development to environmental monitoring, chromatography plays a vital role in various scientific disciplines. Advances in sample preparation technology in chromatography. What are they? Is automated sample processing. So new advancements have led to automated sample preparation and reduced the manual labor and increased the efficiency. And then online sample cleanup in GC, HPLC and other hyphenated techniques. The integrated sample cleanup into the chromatographic workflow streamlines the process and enhances the precision and accuracy. We need not to do manually. So miniaturization and micro extraction. We have seen in the initial slides like cutting edge technologies. A GC can be handled in a hand. Miniaturization, microchips. These are all the new technologies that have emerged into the old and traditional and analytical techniques. The sample preparation techniques are evolving to miniaturization and optimize the extraction processes for improved sensitivity and selectivity, such as microscale liquid chromatography, miniaturized spectrometer sensing devices. So enhanced solid phase extraction also made us more robust in the chromatography system. Then the new technologies I am going to talk, I'm just going to talk about the introductory things. So the flow-based chromatography, flow cytometry is used in the field of life sciences, including chromatography, to analyze and quantify cells or particles in a fluid sample. It is used in chromatography to measure the concentration and purity of a specific analyte. Flow cytometers uses the laser and detectors to measure the size, granularity, and fluorescence of individual cells or particles as they flow through the narrow channel. The detection will be done. So the flow cytometry in chromatography can detect the impurities, contaminants in a sample, ensuring the quality and purity. So it allows the high throughput analysis with the ability to analyze the thousands of cells or particles per second. That is the greatest advantage of flow base. By combining the chromatography with the flow cytometry, researchers can gain the valuable information about the sample composition and before best than the single individual chromatography, leading to better understanding and optimization of separation process. Then paper-based chromatography. So the low-cost paper-based chromatography devices are gaining attention for field applications. These devices usually act by capillary action to separate the compounds and can be used for detecting the contaminants, drugs, and biomarkers in resource-constrained environments. Straight away, we can take that paper-based chromatography and we can do our analysis wherever it is. So the paper-based chromatography for resource-limited settings, this chromatography offers a practical and accessible solution for chemical analysis in resource-limited settings, like providing a cost-effectiveness, portability, and versatile platform for a wide range of analytical applications. So we can take this to anywhere and do wherever we want. So this device uses the capillary action to separate the compounds, and the simplicity and lightweight nature of the paper-based chromatography makes the easy transport and easy handling. Miniaturized chromatography systems. This trend continues with gaining performance. These systems often portability, microfluidic, and chip-based chromatography reduces the solvent consumption, faster analysis times. So the researchers are exploring the applications in point-of-care diagnostics and environmental monitoring. Wherever we want to analyze the environment, we can straight away use all these portable equipments and real-time analysis can be done. Then the high performance liquid chromatography with AI-driven optimization. Researchers have integrated the artificial intelligence algorithms into HPLC systems. These algorithms optimize parameters such as mobile phase composition, gradient profiles, column selection. So the result is improved and separation will be efficient and reduced analysis time. Then coming to the point, quality by design. Implementing the quality by design principles ensures the robust and reliable chromatographic methods. The researchers focus on critical method parameters like risk assessment and process understanding. The QBD in HPLC involves a proactive and systematic approach for the method development and optimization, and control and focusing on understanding and controlling the critical method parameters to ensure that the quality, reliability, and regulatory compliance of the analytical results. Thereby, we can reduce the cost, we can reduce the chemicals, we can reduce the time. So then the advantages of lab automation increases the productivity, efficiency, accuracy, precision, reduced human error, faster turnaround time, standardized and reproducible results can be obtained. Then multi-dimensional chromatography. This is a newer technology. In multi-dimensional chromatography, we can include LC into LC, GC into GC. And this is a powerful approach for separating and analyzing the complex samples by combining multiple chromatographic techniques to enhance the resolution, selectivity, and peak capacity, and enabling the comprehensive characterization of complex mixtures in various analytical applications. So we can improve the resolution, increase the peak capacity, enhance the selectivity, and reduce the matrix effects. Cost can be cut, optimize the resource utilization, enhance the data integrity, flexibility, 24/7 operation can be done. Then coming to the fourth point, mass spectrometry. So you all are aware of mass spectrometry. As we are running late, let me just define spectrometry. So the mass spectrometry refers to the analytical technique used to measure the mass to charge ratio of ions. So process detection and applications, we all are aware of it. Then coming to hyphenated techniques, I will read out clearly because these are the newer techniques. LC-MSMS, liquid chromatography, tandem mass spectrometry. GC into GC-MS, comprehensive two-dimensional gas chromatography mass spectrometry. LC, IMS, MS, liquid chromatography, ion mobility spectrometry, mass spectrometry. LC into LC-MS, comprehensive two-dimensional liquid chromatography, mass spectrometry. Then UPLC, Orbitrap, MS, ultra performance liquid chromatography, Orbitrap, mass spectrometry. These are the new hyphenated techniques, what we are seeing in the new era. Then ion mobility mass spectrometry, IMMS. This is a technique used to separate and analyze the ions based on their size, shape, and charge. So it combines the principles of ion mobility as well as the mass spectrometry. So that is a great advantage in the invention of drug. So it can be utilized in drug discovery, proteomics, and environmental analysis. So it involves the ionization of sample followed by separation of the ions based on their mobility in a gas-filled drift tube. So the separated ions are then detected and analyzed based on their mass to charge ratio using the mass spectrometer and enables the identification and quantification of the ions present in the sample. So the ion mobility mass spectrometry enhances the researchers, what are the advantages are, the researchers have made the significant advancement in IMMS and improving the resolution and selectivity. So the novel drift tube design and advanced algorithms allows for better separation of ions based on their mobility and mass to charge ratio. So the next, single cell mass spectrometry imaging. Here, single cell mass spectrometry imaging is a powerful technique used to analyze the chemical composition of an individual cell. So it combines mass spectrometry, which measures the mass and abundance of the ions, with imaging, which visualizes the spatial distribution of the chemical compound. And SEMSI allows the researchers to map the molecular profiles of the cell in a tissue sample, providing the insights into cellular heterogeneity and the function. So by analyzing the metabolites, lipids, proteins, and other molecules within the individual cell, SA and MSI can help identify the disease biomarkers, study the drug effects at a cellular level. So SEMSI is a highly sensitive and allows for high resolution mapping, but it also requires the specialized equipment and expertise to perform and interpret the data. So this SCMSI enables the spatially resolved metabolomics at the single cell level by combining mass spectrometry with microscopy. Researchers can map metabolite distribution within the individual cell. Thereby, we can assess what exactly it is. And if you can know about the individual cell, you can know about the organ, thereby the patient data. And we can also use this technology in drug discovery. Top-down proteomics with ultra-resolution instruments. The top-down proteomics with ultra-high-resolution instruments is a powerful approach for studying the intact proteins, offering the exceptional mass accuracy and resolution. So it enables the analysis of protein isoforms, post-translational modifications, protein-protein interactions, revolutionizing our understanding of protein complexity and function. So the top-down proteomics analyzes the intact proteins rather than the peptides. So the recent innovation in ultra-high resolution mass spectrometers allows for comprehensive characterization of intact proteins, including post-translation modifications. The advantages of top-down proteomics include high throughput, comprehensive analysis, precise protein identification, insight into protein function. Then the last topic, the spectroscopy. The spectroscopy is a study of absorption and emission of electromagnetic waves by the matter. So that definition includes the study of the interaction between the particles such as electrons, protons, and ions. So advanced technology in spectroscopy is like two-dimensional spectroscopy, ultra-fast spectroscopy. So in two-dimensional provides the enhanced spectral resolution, elucidating the complex molecular structures and dynamics. In ultra-fast the process on femtoseconds to picoseconds time scales, crucial for investigating the photophysical and photochemical dynamics. And in single molecular spectroscopy, it detects and characterizes the individual molecule, offering the insight into molecular heterogeneity and dynamics. In super resolution spectroscopy, surpasses the diffraction limit of conventional optical microscopy and visualizing molecular structures and dynamics with nanometer scale resolution. In mass spectrometry imaging, maps molecular distribution within the samples, facilitating the research in biology, medicine, and pharmacology. The terahertz spectroscopy probe molecular vibrations and properties using terahertz frequency radiation with applications in pharmaceuticals, materials, and security screening. So, coming to the last topic, applications of artificial intelligence in analytical science. Machine learning, it utilizes the algorithms to analyze the data and makes the prediction. Data processing, efficiently processing the large volumes of the complex data. Automation, automating the respective task for enhanced productivity. Pattern recognition, identifying the patterns and trends in data for insights. So artificial intelligence can be utilized perfectly in drug discovery and development, environmental analysis, food and beverages, forensic analysis, quality control in manufacturing, whatnot, in arts, science, engineering. In every world, AI has emerged like anything in recent times. So applications of machine learning. So machine learning has revolutionized the field of analytical sciences by providing the powerful tools to analyze and interpret the complex data sets. And one of the key applications of machine learning in analytical science is in development of predictive models for chemical and biological analysis. And by training the algorithms on large database, researchers can predict the chemical reactions, biological process, and material properties with remarkable accuracy. The only problem with this technology is initially we need to have so much of information, and that should be dumped into the system, and then it effectively identifies what it is exactly. So initially we have to collect lots and lots of data and give the inputs to the machine. Then challenges and future prospects. Novel methodologies and techniques, interdisciplinary collaboration, data availability and quality, data privacy and security, funding and resources, and computational resources are the limitations or challenges of the AI-based technologies. Then challenges and limitations of the analytical innovative methods. The regulatory hurdles, like compliance with stringent regulatory requirements, because day by day newer and newer technologies are emerging. Portability, miniaturization, personal medication, all these things have emerged. So to get approvals on all these things by our regulatory authorities is a big hurdle. It takes time, though the new technologies are emerging. Then technology complexity, integration with multiple kinds of technologies, methods, new devices. So interoperability of advanced technologies and integration is again a big complex system. And then data security. The data, whatever is obtained and utilized for the newer technologies in AIs and even in analytical systems. Data security is an important thing and a considerable issue. And then privacy of the sensitive data is going to be a big thing. How to overcome the problems? So we can implement, just a minute. The ways to overcome the challenges in analytical field. So we can implement the automation and AI in drug discovery, leverage the blockchain for security supply chain management, utilize the cloud-based platforms for data storage and analysis, embrace the telemedicine and digital health solutions for remote patient care, invest in VR and AR technologies for medical training and education, explore 3D printing in pharmaceutical manufacturing, develop the wearable devices for real-time patient monitoring, utilize the genetic engineering for personalized medicine, enhance the cybersecurity measures for data protection, utilize the big data analytics for improved patient outcomes. So to put it into a nutshell, what I have tried to tell you is we have so many newer technologies and we have conventional way of analysis like chromatography, spectroscopy, and other techniques. So these new technologies, AI, IoT, portability, or new devices, AR, VR, and all these technologies have emerged into these olden technologies and made the mankind survival very easy. At the same time, it is also a complex thing. Why I have chosen this topic is I have not chosen the single topic and I have not explained in deeper way, because nowadays we have got so many technologies into the world, but in academics, usually we do not try to know all these techniques, because what is the ultimate goal of a teacher is what is our syllabus, whether we have taught, whether we have finished it or not. But so they has come for all of us in teaching to improve us. Why? Because the world is changing very fast. We have to adapt to the new technologies. We have to know the new technologies. We have to get ready to do the multi-dimensional research, and the personalized medicine is hitting soon. So we have to develop new methods, new technologies, cost-effective technologies. Requirement is increasing. So we must be able to do the research. Then the portable devices and robotics has entered into our system. So we have to know about these technologies. Non-invasive devices have increased. NEP 2020 revolutionary changes in education system is going to come. Indian government is encouraging us. If we have an idea which is useful to the society, they are encouraging with lot many funds. So it's you develop your skills, adopt the new technologies, face the challenges, try to know much about the outer world, think out of the box. Don't think only about your syllabus prescribed by PCI or our universities. Beyond that, you have to know, and whatever is known by you should be taught to the students, even though it is not specific in the syllabus. So to put it in nutshell, develop your leadership skills, do a SWOT analysis of yourself, where you are good, where you are weak, keep your health in a very good condition, take care of yourself, know the technology, be bold and fear not, face the new challenges. Let's face all these technologies, and thank you so much. Then we conclude our topic with a famous quote of Albert Einstein: intellectual growth should start at birth and cease only at death. Thank you all.
Thank you, madam.
Over to you, Sharma ji.
Madam, thank you, madam. And if there are any queries from our participants. So participants can unmute yourself and ask if there are any queries to Madam. Madam, I hope there are no.
It means it is everything is understood or nothing is understood. Okay. Yes, please.
Any questions from participants? I hope everybody has understood, Madam.
Let's hope like that.
So, thank you, Madam, for your valuable and informative talk on advances in analytical techniques, Madam, and enlightening our participants with the cutting edge technologies such as image technologies, biosensors, bioanalytical methods, microfluidics, chromatographic techniques, spectroscopic techniques, and multidimensional techniques. And you have given an excellent presentation, Madam, in spite of your busy schedule. So thank you for accepting our invitation for the benefit of our faculty fraternity, Madam. And as you have started your presentation, the secret of getting ahead is getting started. And we have started with an FDP series, Madam, and it will be continued. And we want your support in the future also. And to all the participants also, thank you for your valuable time. And do support us in the future also, dear participants. And thank you so much for your help, Madam. Thank you
So much, sir. It's my pleasure to be part of your program. Thank you.
Madam, I request our assistant professor, Kalyani Madam, to deliver the vote of thanks. Kalyani Madam, please continue.
Unmute yourself, Kalyani.
Okay, ma'am. Good afternoon to everyone. Myself, K. Kalyani, Assistant Professor, Department of Pharmaceutical Analysis, Sri Vasavi Institute of Pharmaceutical Sciences, Tadepalligudem. I am here to propose a vote of thanks regarding a 5 days comprehensive FDP on innovative methods in analytical sciences, which is organized by Sri Vasavi Institute of Pharmaceutical Sciences since from March 11 to 15. My sincere thanks to resource persons who delivered valuable information regarding various analytical techniques. Dr. A. Krishna Manjari Pawar Madam, Associate Professor, Andhra University, Visakhapatnam, and Dr. M. Kiranmayi Ma'am, Professor and Principal of St. Paul's College of Pharmacy, Hyderabad. Dr. A. Lakshman Rao Sir, Professor and Principal of VVIPS, Gudlavalleru, and Dr. M. Murali Krishna Sir, Associate Professor, Andhra University, Vizag, and Dr. K. Ravikumar Sir, Professor and Vice Principal of Hindu College of Pharmacy, Guntur. My deep gratitude to all the speakers for their in-depth explanation and detailed information about innovative methods in analytical sciences, which has enabled all our participants to understand the concepts excellently. My sincere thanks to all the participants for attending this 5 days online FTP conducted by Sri Vasavi Institute of Pharmaceutical Sciences. I am thankful to our management members, C.H. Apparao Sir, and Principal of Sri Vasavi Institute of Pharmaceutical Sciences, Dr. V. Bhaskaraju Sir, and Organizing Secretary of this FTP, Mr. K.S. Suman Sir, and all the organizing committee members, Mrs. Amini Kumari Ma'am, and Sharma sir for their organization and coordination for smooth conducting of this online FDP. This FDP provides a great opportunity to learn a great insights about various innovative methods in analytical sciences. Once again, thanks to all the resource persons and participants. Thank you all.
So thank you, participants, and thank you, madam, for your support.
Thank you to all the participants. So we will share the feedback link within 5 minutes in the WhatsApp group. Thank you.
Registration certificates గురించి కూడా చెప్పండి.
So all the participants will get certificates. We will share the certificates within 1 week. Thank you.